Chapter 7 now covers the membranes. Remember we talked about we have membranes around our organelles, we have a membrane on the outside of the cell, the nucleus, so on. So there's special things that these membranes do to perform their function, which is basically they're the gate. They allow some things to go in and out and some things not to go in and out. And they have a special structure and allows them to do that. So life at the edge, yeah. The plasma membrane, remember, is the boundary that separates the living cells from the surroundings, okay? So that when you can actually see the plasma membrane when you look under a microscope. The plasma membrane exhibits what they call selective permeability. In other words, some things can come in, some things can't, okay? And it's important because you got to keep stuff inside and you got to keep stuff out. But remember, organelles have jobs to do and they need certain materials. So the plasma membrane is what allows the materials in. And then once they make something, it needs to go out wherever it's supposed to go. Same kind of deal. Okay. Here's an example. This looks, here you can see the plasma membrane, the layers and stuff. These are little water molecules. Okay. But kind of funky looking, huh? Okay. So cellular membranes, they're fluid, okay? They have like a mosaic. They have a mixture of lipids. Remember, lipids are fats and proteins, okay? They call them phospholipids, all right? And they're the most abundant lipid in the plasma membrane. They line up so they have opposite ends, and again, I'll show you that here in a minute. But they have one end that's hydrophilic and one end that's hydrophobic, one end that likes water and one end that doesn't like water. Okay, they're amphiphilic. In other words, they have two different regions, and I just talked about that. Okay, anytime you see this word in the front, like there's an amphoteric, water can be amphoteric. It means it can be either an acid or a base. There's all kinds of things, but this just means can do both things. Okay, so when you look at a phospholipid, one end is hydrophobic, doesn't like water, and one end is hydrophilic, and it uses this to its advantage to get things in and out. Here you go. Excuse me, sorry. Here's our hydrophilic, the one that likes the water, and down here, this would be another layer of that. So they line up, there's two of them, and they line up basically tail to tail. So here's the head up here. This is the outside and the inside. And then these are the tails. And you can see they look, here's the head if you want to look more molecular. And again, this end likes water, this end not so much. Okay. Okay. They also have protein globs. And the protein globs, remember we talked about enzymes and they have like a site that they go into? Well, the proteins help move kind of bigger stuff in and out. Okay, they attach to it and they kind of slide it in, and or they attach it out here and slide it out. So we have this phospholipid layer, and you can see, and then we have these big old proteins in there that that help move bigger things in. Okay, the small things can go in and out easily with this layer, but sometimes we need bigger things, and so that's what the proteins embedded into the membranes help do. Okay, as the temperatures cool, the membranes are going to switch, just like most things, from a fluid to a solid state. Okay, so the temperature of the membrane solidifies. It depends on, because remember with the lipids, if it's a saturated one, it's going to be a solid at room temperature. If it's a unsaturated one, and that just means it has double bonds or triple bonds, or more than one of those, then it's going to be a liquid at room temperature. So it depends on those lipids, and again, that depends on what needs to come into the cell and what needs to go out of the cell. Okay, membranes rich in unsaturated, like I just said, they're going to be more fluid because they're not solid. Okay, if they're solid at room temperature, like saturated fatty acids, okay, they're not going to be as fluid. Excuse me. Membranes must be fluid to work properly. Okay, so again, the unsaturated are going to work better because they're like salad oil. Okay, and again, when we talked about unsaturated and saturated fats, your plants, your vegetable oils, your olive oil, canola oil, all those are liquids at room temperature, whereas the animal fats like butter, Crisco, those things, they're solids. So in a membrane, works the same. You want the more unsaturated. Okay, the oh, sorry, you guys. The sterile cholesterol has different effects on membrane fluidity at different temperatures. Again, cholesterol is one of the groups of lipids. Okay, at a warm temperature, cholesterol restrains movement of the phospholipids. Warm temperatures such as 37 degrees Celsius, that's our normal body temperatures. That's like 98.6, which most of us know our body temperatures pretty well now, because I'm sure if you're like me, you've had your body temperature taken a lot when you go places. Okay, as temperatures cool, it maintains the fluidity, preventing tight packaging. So it wants it to be kind of spread out, and you'll see there's sometimes cholesterol molecules intermingled in the phospholipids. And again, that's just to help to space it out a little bit and allows better channels for things to move in and out. Okay, here we got it. Oops, sorry. Here we got our stuff here. Okay, if it's really tightly packed, here's your saturated. Okay, this would be a membrane that doesn't allow a lot of things to move in and out. So what happens is cholesterol kind of moves in here, and it helps, you can see, unpack things a little bit. So there's some channels for more things to go in and out. And again, it depends on the membrane, it depends on the cell. The more cholesterol you can have in there. So we always hear is cholesterol is bad, 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 bad. It's not always bad. You know, sometimes we need it. We do need some cholesterol in our body to help with things. And this would be one case in our membranes in our cells to make sure that we can allow things to move in and out. Okay. The variations in the composition, they think is an evolutionary thing. It's allowed us to adapt to certain environmental conditions. Okay. It happened when the temperature changes, because we've got organisms that live in extreme heat, extreme cold. You know, we've got this in the variety of everything in between. So they think, you know, the organisms that were allowed to change their composition are the ones that survived better, and obviously they reproduced, and the ones that didn't died out. So they think that this change in this membrane composition is an evolutionary thing, like survival of the fittest. The ones that adapted their lipid composition in their cell membranes survived better. They had more offspring, and then eventually the other composition, they didn't make it and they passed away. And I'm not just talking about for people cells, you guys. I'm talking about all organisms. Okay. All right. Membrane is is a collage. Okay. There's all kinds of different proteins. Some of them are grouped together and they're embedded in that fluid. And again, they're help move bigger things in and out. Okay. They're embedded in that matrix of the lipid bilayer, that, you know, that phospholipid that's up and down where they together, because there's two of them together. And then we have these globs of proteins embedded in them. Okay. The protein determines most of the membrane specific functions. Okay. Because all of the membranes are going to be similar with the phospholipid, the two phospholipid layers, but the proteins are going to determine what really can come in and out. Okay. There's 6 major functions of those membrane proteins, we'll call them. One is to transport, move things in and out of that cell. Okay. If something's made in the cell, it needs to go somewhere. Let's get it out and get it where it needs to go. And if there's something that needs to come in, same deal. Okay. The enzyme activity. Okay. If you need some enzymes in there, like your mitochondria needs all kinds of enzymes to perform its job as far as making us ATP. We need to get some enzymes in there. Let's do it. Okay. They actually send signals throughout. Okay. So signal transduction, they send information around in that cell. Cell recognition. Okay. Some cells need to be together. I mean, when you put a bunch of cells with the same function together, you have a tissue. Okay. And then you put a bunch of tissues together, you have an organ. Well, those cells need to be together. You have to recognize each other. Intercell joining, that's the next step after cell recognition. If they're supposed to be together, now they're going to join together, and that comes from those proteins. And then if they need to attach to the cytoskeleton, okay, the cell skeleton, and then extracellular matrix, all of those things, again, kind of the next step after they join. Are they there? Are they there to help out? Are they going to provide structure, that kind of stuff? So proteins have a lot of function in the membranes. Okay. The membranes, again, have distinct inside and outside faces. The the distribution of those proteins, remember, determines what that membrane can do. And so it's really determined by two things. The endoplasmic reticulum is what helps build the membranes, okay? Not only its own membranes, because it has membranes, but the nuclear membrane, the mitochondrial membrane, the cell membrane, all the membranes inside the cell are built by the endoplasmic reticulum. They're sent to the Golgi apparatus. And remember, the Golgi apparatus is the shipping and the packaging. Well, it modifies it then to, it's like the last part that really makes it, perfect, I guess. Quality control, I guess, if you want to call it that. So they make all these membranes in the endoplasmic reticulum, and then they're sent to the Golgi apparatus, or the Golgi, whichever way you want to say it. And then they perfect it, make sure that it's ready to go, and then the membrane's sent where it needs to be. Okay, so here we have, you can get this, and these pictures are in your book too, but here's a membrane, we've got parts coming in. Here's a protein, and remember I told you it has to get certain things in, so it's going to close around it, and then it's going to form what they call a vesicle. Okay, these vesicles can allow things to either come in or go out. Okay, so here's one that's formed in the ER, it's sent here, and it's finally packaged up the way it needs to go. Then what happens? Okay, now it's ready. I've got these little particles, these proteins, whatever they are, whatever they need to be. We're going to package them up in a vesicle. The vesicle is going to come up here, and then it's going to say, hey, I'm at the membrane now. Open up, and do-do-do-do, away they go. And now these guys are in the bloodstream, and they'll go wherever they need to go. Okay, so this happens. These are just the Golgi here. They don't show the endoplasmic reticulum. Kind of the same deal, but usually the endoplasmic reticulum makes them, sends them here to get modified, and then they're secreted out. Okay. All right. So the membrane structure results in what they call selective permeability. Okay. Some things can come in, some things can go out. Not everything can leave, not everything can come in. Okay. So a cell must exchange materials. It has to. It has to get rid of stuff. It has to take things in. And that is controlled, again, by the plasma membrane. We talked about that. The plasma membrane is like the security gate, okay? And you have to check in with it, and then it allow things in and allow things out, okay? The plasma membranes are selectively permeable, resulting in the cell's molecular traffic. It's a fancy way, again, of saying they're like a gate. They allow things in, they allow things out, but not everything, okay? If it was just easy in and out, I mean, you'd start losing your organelles. The small organelles like the lysosomes and the periosteums, they would just like sneak out. Can't allow that to happen. They got to stay inside. So that's what the plasma membrane does. Okay, that layer, that the lipid bilayer with, your two ends, your hydrophobic, the one that doesn't like water, that's going to be nonpolar. And then they're going to be attracted to the hydrocarbons, the oils, the cholesterol, okay? They can dissolve in the lipid bilayer and pass through the membrane quickly, okay? So if it's hydrophilic, okay, and that's going to be ions, that's going to be your polar molecules. Hydrophilic meaning they like water, they don't cross through the membrane as easily. So really your hydrocarbons, like your carbohydrates and things like that, those are the ones that are going to pass through. And then the other ones aren't, okay? And then again, you have your transport proteins that allow the passage of the hydrophilic, the ones that they allow some of them, like I said, some of them, if you just go normal, they can't go through. But the hydrophilic, those would be your polar ones and your ionic ones. But the transport proteins, they kind of overstep the bounds. If it was just the phosphobiolipid, these guys wouldn't be able to get in. But the transport proteins kind of step in and allow, like they serve as an escort to let things in and out, okay? Some of the transport proteins are called channel proteins. They actually form a channel that they can swim on through, they can tunnel in. And again, certain molecules, certain ions, and so on. Okay, we're going to quit there because I'm just about done. Remember, this is about just the membranes, and these are the membranes around the cell, in the organelles, and everything. So this is the end of part 1.